Good morning. So I'm really honored to be asked to present. I wanted to point out that although I'm in the beginning here of the presentation, I think there are much more interesting speakers after me, because what I'm going to tell you about is the kind of news that just broke this past week. This is a presentation I haven't really done before, so bear with me as we go through this. I've put a lot of video into the slides, and hopefully they'll work without a hitch. But you'll hear from folks like Dr. George Runger, who's head of our Biomedical Informatics department here at ASU, some companies that are doing amazing, fantastic things, helping us understand health from a wearable devices to integrate and analyze data perspective, as well as kind of the support from the city that we're getting, which is fantastic. So today I thought we'd talk about how technology leads these breakthroughs, right? So we're talking about hacking the human. If you go to the next slide. Systems Imagination is a company that we started about 5 years ago, focused on how can we apply artificial intelligence and other data analytic methodologies to health data that's already out there. In this case, biomedical data that helps us understand what's happening in biological systems. And we worked with pharmaceutical companies to help them understand what their drugs were doing inside of humans. Next slide. This led to the formation of a company called Systems Oncology, which is one of the sponsors today. I encourage you to talk to some of the folks who will be here this afternoon. Basically, the idea was, if we could take all this knowledge that we're mining, could we then identify new potential therapeutics which could be rapidly passed through the drug development process to get these drugs to cancer patients. And that work is going on right now up in Scottsdale. It's a fantastic company. It's about 2 years old, and it's going to be a great success story for Arizona, which is really what we're trying to achieve, is get more of these drugs to patients faster because we know more about biology now. Next slide. And we're forming this kind of movement, we'll call it, called expansive AI. It's the concept of, if you could create all the pieces that are necessary for any scientist or physician to look at any disease and say, I know exactly what's wrong with that patient, and therefore I know what the armamentarium of drugs or therapies are that we should use, and I'm using all the technology around me. Well, that's actually machines learning to help us make decisions, and that's where we're headed. And I think Arizona in particular could be a place where we could do that kind of work. So now let me switch gears. I'm gonna head right into this past week's controversy of hacking the human. This is true hacking the human. It's gene editing. Some of you have probably seen the news in the papers. I'll start with this, which is, thinking about, where do these ideas come from? Why do we search for innovation? Why do we try and achieve breakthroughs? The right side of the screen is a list of selected top 10 discoveries or breakthroughs as noted by Science magazine over the past 10 years. And you'll see they're very immersed in genes, genomics, gene editing, diseases like HIV, and of course, artificial intelligence. But there's a reason why startups always have foosball tables. And I think it's because there's this inherent need to kind of compete amongst humans. And when you're competing, you're starting to think together about a problem. Right, and next slide. And just 2 years ago, a company that's now part of Google called DeepMind created something called AlphaGo that beat a human at one of the most complicated games out there called Go. And that kind of made me think about, okay, where do these ideas come from, and will machines ever get there to be able to help us come up with breakthroughs to hack the human? Well, this next slide is the actual Chinese researcher who claims to have edited human embryos and put in a gene into newborn infants who are now supposedly resistant to HIV infection. And I thought it would be very important to hear what he says, because we want to understand what causes these things, what makes people want to innovate, and what are the boundaries that we need to think about as people apply technologies that are coming very fast at us, especially with things like artificial intelligence. So,
two beautiful little Chinese girls named Lulu and Lala came crying into the world as healthy as any other babies a few weeks ago. The girls are home now with their mom Grace and dad, Mark. Grace started her pregnancy by regular IVF, with one difference. Right after we sent her husband's sperm into her egg, we also sent in a little bit of protein and instruction for a gene surgery. When Lulu and Lala was just a single cell, this surgery removed the doorway through which HIV entered to infect people. A few days later, before returning Lulu and Lala to Grace's womb, we checked how the gene surgery went by whole genome sequencing. The result indicated that the surgery worked safely as intended. Grace's pregnancy was normal, which we monitored closely by ultrasound and blood tests. After birth, we again deep sequencing Lulu and Lala's whole genome. This verified the gene surgery worked safely. No gene was changed except the one to prevent HIV infection. The girls are safe, healthy as any other babies. When Mark saw her daughter for the first time, he said that he never thought he could be a father. No, he has found a reason to live, a reason to work, a purpose. You see, Mark has HIV. Discrimination in many developing countries makes the virus worse. Employer fire people like Mark. Doctor deny medical care, and even falsely sterilize a woman. Mark and Grace couldn't bear to bring a child into that world of fear. Mark's words told me something I didn't fully appreciate. A gene surgery that could save a child from a lethal genetic disease like cystic fibrosis or from a life-threatening infection like HIV. It doesn't just give that little boy or girl an equal chance at a healthy life. We heal a whole family. As a father of two girls, I can't think of a gift more beautiful and wholesome for the society than giving another couple a chance to start a loving family. The media heaped panic about Louise Brown's birth as the first IVF baby. But for 40 years, recognition and morals has developed together with IVF, ensuring only therapeutic application to have more than 8 million children came into this world. Gene surgery is another IVF advancement, and is only meant to help a small number of families. For a few children, early gene surgery may be the only viable way to heal an inheritable disease and prevent a lifetime suffering. We hope you have the mercy for them. Their parents don't want a desired baby, just a child who won't suffer from a disease which medicine cannot prevent. Gene surgery is and should remain a technology for healing. Enhancing IQ or selecting hair or eye color is not what a loving papa does. That should be banned. I understand my work will be controversial, but I believe family need this technology, and I'm willing to take the criticism for them. So you can learn more about our more.
Okay, so that is his video that he released Sunday before this human gene editing conference in China took place this week. The next slide is a video of one of the inventors of CRISPR-Cas9, which is the technology that he used to edit these embryos. And this was an interview with 60 Minutes a couple weeks before this conference.
We were speaking just recently with a scientist who hopes to use CRISPR to prevent disease. Do you think it's better to treat the disease or try to prevent the disease?
Treating disease, I think, is certainly a good approach because we know there is a problem and we can go and try to fix it. Now, preventing disease, I think, is a little more complicated because it depends on how we're trying to prevent the disease. Some of them may have undesirable side effects that we don't fully understand. I'll give you one example. One way to prevent HIV infection is to remove a gene called CCR5 from people. So those people will not get infected by HIV virus, which you may think is a great thing. These people are immune from HIV. But the caveat is that if a person doesn't have CCR5, their risk for West Nile virus increases significantly.
For West Nile virus.
For West Nile virus. So if you prevent the disease by removing CCR5, you are also installing a new risk to those people. And that's where I think we need to be very thoughtful and careful.
And this would be something that would be passed on from generation to generation. If you tinker with the embryo,
right,
that person will then pass that altered
to their children,
genetic makeup onto their children.
Right.
Is that what makes this so controversial?
I think that certainly is something that's really important to think about, because you're altering the human germline.
And the germline
The germline is our DNA, and it can get passed on to the next generation. That's definitely one thing that makes it controversial. But the second thing is, because we don't understand biology, we don't know what the consequences of those changes will be. And that's why we don't want to edit right now. We shouldn't.
So you heard both the person
we were speaking
pushing the frontiers and the person who invented this technology. They're both saying that this is technology that has arrived, and we now need to understand biology better. We need to understand what's actually behind these changes and what the long-term consequences might be if we use this technology. Jennifer Doudna is one of the other inventors of this technology. She also stated, this is very disturbing, stated this week at the conference, if you could type through. The conference itself, which consisted of experts in the field, including David Baltimore, one of the Nobel laureates, also said this is technology we should not use until we figure out a way to really control it. Although this conference did say, hey, it's coming and we need a process to figure out how to enable trials that are controlled. So I think, one more slide there, the Chinese government, the vice minister of science and technology also said, hey, laws were actually broken here, and this is shocking and unacceptable work. But I want to try and pull away the ethical, social, legal implications, because those are things that could take many days in another conference. I wanted to actually look at the data, right? So the question was, okay, CCR5 was mentioned. Feng Zhang said, we don't know enough about the biology. So that's our job, right? As data analysts, we look at data, we perform bioinformatics, we look at, and I'm not going to read through this because it's too much information here, just to point out that you can look and see that CCR5 is well studied in HIV. There were decades of research on it. CCR5, as it relates to West Nile virus, is not well studied. But because we know a lot about how HIV works to infect cells, this researcher was able to take a variant that's commonly found in Northern European populations and edit that into the embryos. That's what he did. What Feng Zhang is talking about is taking that gene, CCR5, and disabling it. So there's some slight differences here of what we're talking about, but the controversy has been so hyped up that it's very hard to just kind of focus on the data. But we think that if you actually look at the data, the experiment that he performed was a rational scientific experiment. Now, he ignored a lot of things that he shouldn't have, in my opinion, but the far right part of this panel is highlighted in a red box. 60% of the Chinese population, according to this recent survey, believe gene editing should be legal, and if you can solve problems like HIV using this technology, it should be used. So we have a lot of work to do as a society. We have a lot of work to do as a scientific community to think about how this works. But the main point is the data is out there. There are going to be folks mining that data, and the question is, if we can edit any gene in our genomes, what do we do? How do we kind of focus ourselves on the ones that should be edited? So, you know, there are great stories, on the other hand, of gene therapy. Gene therapy is how you deliver genes that have been modified, right? In this case, for example, RPE65 is a gene that's well known and well studied, causes, when it's in a mutated form, blindness, retinal dystrophy. There's a company called Spark Therapeutics. Their therapy was just approved at the end of last year. They basically inject this normal wild-type gene, RPE65, into the eyes of people with this mutation who are blind, and now many of them can see. I mean, it's an amazing story. It's a great story, but it also costs $850,000 per treatment. So this is only something that affects 1,000 to 2,000 people in this country, and this is really the only place where you're going to get that kind of therapy. So we have to think, you know, how do we decide and how do we look at the data in a way that helps rationally focus us around how to use this kind of disruptive technology? Because we can affect every one of the 20,000 genes, but maybe we should focus on the ones that are leading causes of death and have, for example, single gene to disease phenotype relationships. Next slide. So I have 3 more slides here. I'm just gonna, let's skip this one actually. We're gonna skip this one. Pharmaceutical companies are, and talk about the last 2 slides. I think people are worried about the left side of this panel. Aldous Huxley's Brave New World, published in 1931. If you haven't read it, I'm surprised. It's a dystopian future where embryos are edited and then the resulting humans are classed based on how they've been edited. This came out right after Ford invented the assembly line, so there's a before Ford and an after Ford period. It's a very dystopian view, and people, when they read this, thought, oh my gosh, maybe we're headed that way. And then the Second World War happened, eugenics, all that kind of stuff. Does anybody know who the person on the right is? That's not a drinking device, by the way, or a smoking device. She's a healthy 40-year-old mother of 2 in England. That's Louise Brown. She's the first human conceived by IVF. Okay, and as He mentioned in his video, 8 million babies have been born from IVF. Back then, it was a humongous controversy. People were thinking we were going to have population explosion, lots of problems. Yet we were, I think, smart enough back then to figure out how to handle this new technology and have it solve medical conditions in the right way. The kind of Brave New World did not happen in that case. This last slide is, if you could just page through, yeah. I think the next couple of talks, you're going to hear about 3 things that are coming next. Ways in which we can leverage our genome sequence, because there is nothing more hacking the human than your own genome. Integration of data between devices and things that are going to come to us directly as genomic data. And if you could click on that guy to just play. And then the final thing is, what's exciting to me, tying this back to the first slide, AI systems that actually can now imagine, right? So what you're seeing here is a video from the same company that built that AlphaGo, that built the Go champion. They just published this past summer. They can now give their neural networks just single, maybe 2-3 images, single 2-dimensional pictures, and the neural network will create the entire 3-dimensional space. So in other words, it can perceive different views that it's never seen before based on what these top images are of 2-dimensional space. And you think about that, the only way you can do that is if the system, in this case the neural network, is imagining the space. So we're at a stage now where AI is going to imagine things for us, and that's why I think things like, gee, what should we work on with gene editing? How should we hack the human? We got to get ahead of that before these systems do, right? Because it's going to be a competition, and it's going to be good for us. But I think we'll end up in a stage just like IVF 40 years ago, where we're going to have the right regulation in place to kind of control that. So with that, I'll hand it back to Rick, and thank you very much.